So what is cholesterol? Anybody who knows what is cholesterol? We will discuss its metabolism today, right? The synthesis of cholesterol and the regulation of cholesterol synthesis. What are steroids and what is sterol ring? Anybody who knows? What are steroids and sterol ring? The formula is C27 H45 OH, or you can write C27 H46 O, right? And it is represented by CEPP, cyclopentano perhydro phenanthrene nucleus, right? And it has 4 rings, 3 6-membered rings and 1 5-membered rings with a side chain containing methyl group, right? And it is basically found in animal products. So the name is animal sterol, right? And it is amphipathic in nature, right? What does it indicate? It contains both polar and non-polar groups. It has both charges, it is polar as well as non-polar, and the formula is C27H45OH, cyclopentano perhydrophenanthrene nucleus, and 1, 2, 3. The 3 rings are 6-membered, 4th ring is 5-membered, and there is a side chain, right? And exclusively found in animals, so the name is animal sterol, and made of a steroid nucleus. I drawed it, known as cyclopentano perhydrophenanthrene ring or nucleus, right? And it is a major component of cell membrane and lipoproteins, right? And suppose there is a adult man, that is a normal adult man, that is weighing only 70 kg, right? So how much amount of cholesterol it will contain? It is equal to approximately 140 gram. So how will you calculate? 2 gram per kg body weight. 2 gram per kg body weight, right? So suppose someone is 80 kg, right? 2 gram per kg body weight. And amphipathic in nature, I told, because it contains both polar and non-polar groups, right? And what are the functions of cholesterol? It is good or bad? It is good or bad? Tell me. Good. And what are the functions? First discuss. It is structural component of cell membrane, right? As phospholipids, right? And insulating cover for transmission of electrical pulses in nervous system, right? And has role in membrane structure and function. Beside this, it is also a component of vitamin D, right? Sex, steroid hormones, cortical hormones, and synthesis of bile, it plays a role, right? And fatty acids are transported to liver as cholesterol esters for oxidation, right? So the functions are there. I told 1 gram of cholesterol is synthesized per day in adults. Normally, 1 gram is synthesized by body daily, right? But where it is majorly synthesized? Tell me the organ where cholesterol is synthesized. The major organ is liver. Number 1 is liver. Number 2 is intestines. Anything left? And where which fraction of the cell cholesterol synthesis take place? Anybody? Which fraction of the cell cholesterol synthesis take place? Cytosolic or mitochondrial? Any guesses? Anushka, can you guess? Sina, can you guess? Which fraction of the cell? Cytosolic. Cytosolic fraction of the cell. It happens in cytosolic fraction of the cell, right? And the steps are very difficult. Big diagram. Just to overview, then we'll discuss. This is the steps. It appears horrible to you, but we will make it easy with mnemonics. And it is very important, 4-5 marks question will come, right? But I will try to simplify it. So let's get rooted again. Don't get depressed. Just see, I will make it easy for you. Mnemonic बता दूं, बोलो. Request करो ना फिर, please बोलो. Yes. The mnemonic is high miscellaneous. High miscellaneous. Right? There are basic 5 steps first. 1, 2, 3, 4, and 5. High for synthesis of HMG CoA. High, first letter, right? Then this is 2, synthesis of mevalonate. Right? Then come I for third. Isoprenoid, synthesis of isoprenoid units. Right? Then comes S for fourth step, synthesis of squalene. Right? Then C for fifth step, synthesis of proper cholesterol. High miscellaneous or high mis. H for synthesis of HMG CoA, M for synthesis of mevalonate, then I for synthesis of isoprenoid units, then S for synthesis of squalene, and C final for cholesterol. These are the 5 major steps, right? And this is the broader picture. Now I will also break down it into smaller picture. Take screenshot first. Please take screenshot. It will help you. Afros le le. Then the first is synthesis of HMG CoA, right? How will you synthesize HMG CoA? Can anybody recall ketone body synthesis? Ketone body synthesis. All of you are coming blank. That's the problem. How will you synthesize ketone bodies? What is the raw material for the synthesis of ketone bodies? Anybody? Building block or the single brick from which we can synthesize ketone bodies? Acetyl CoA. We synthesize ketone bodies from acetyl CoA. We synthesize cholesterol from acetyl CoA. But the difference is that this cholesterol synthesis happens in cytosol, and this happens in ketone body synthesis take place in mitochondria. First is the major difference, right? The cholesterol synthesis is for C for cholesterol synthesis, C for cytosol. Ketone body synthesis is take place in mitochondria. But the first three steps are same for the synthesis of HMG CoA. I will break down. First of all, acetyl CoA plus acetyl CoA. What will you get? Acetyl CoA. Again, another molecule of acetyl CoA is added. We will get HMG CoA or beta hydroxy, beta methyl glutarate CoA. Right? The first step, write the mnemonic is high miscellaneous. We starting from high. High is HMG CoA, right? But HMG CoA requires these first three steps. These are same as ketone body synthesis, but this cholesterol synthesis is taking place in cytosol, but this ketone body synthesis takes place in mitochondria. Right? So first three steps, one molecule of acetyl CoA combines with another molecule of acetyl CoA will get acetyl CoA. Then again one molecule of acetyl CoA is added and will get our HMG CoA. The other name is beta hydroxy, beta methyl glutarate CoA or HMG CoA. Right? Now we have the first product, that is HMG CoA. Right? And remember, for the synthesis of cholesterol, you need some things. Raw materials, reducing energy is provided by ATP, right? Reducing equivalents are provided by NADPH, and it takes place in cytosol, and the carbon atoms are derived from acetyl CoA. It is a big ring, na? It is a big ring. Cyclopentano per hydrophenon 3 nucleus. 3 6-membered rings and 1 5-membered ring is there, right? And it contains carbon atoms. The carbon atoms are derived from this acetyl CoA, right? See, from acetyl CoA we are forming acetose, then HMG CoA. And this HMG CoA is the precursor for the steps, right? And the carbon atoms are coming from acetyl CoA, right? And reducing equivalents are provided by NADPH. What is the source of NADPH? HMP shunt. Energy is provided by ATP, right? Now the first step is clear, the synthesis of HMG CoA. Synthesis of HMG CoA, and how many carbons are there in HMG CoA? 6. It is a 6 carbon compound. We'll start from acetyl CoA. We add another molecule acetyl CoA, will get acetoacetyl CoA. Then another molecule acetyl CoA is added, we will get our HMG CoA, right? The first product is now formed, right? Now we'll form mevalonate. How will you form it? It is again a 6 carbon compound. Now we have HMG CoA, it is converted to mevalonate. Both are 6 carbon and it is also a 6 carbon, right? How will you move into this? Here comes the role of enzyme HMG CoA reductase, and it is a regulatory enzyme for cholesterol synthesis, right? And we have two fractions of this cell HMG CoA reductase. One is cytosolic and one is mitochondrial. Mitochondria will play a role in ketone body synthesis, and this cytosolic will play a role in our cholesterol synthesis. So we have this cytosolic because we are going for cholesterol synthesis, right? So this is cholesterol synthesis, and cholesterol synthesis takes place in cytosol. So we have this cytosolic fraction of this enzyme HMG CoA reductase. From HMG CoA will form mevalonate, that is 6-carbon compound, right? So I started the with mnemonic high Mbbs, high for HMG CoA. HMG CoA is got from which compounds? Two molecules of acetyl CoA combined to form acetoacetyl CoA. Then another molecule acetyl CoA is added, it will form HMG CoA, right? And high Mbbs M for our second compound, that is mevalonate. And the enzyme is HMG CoA reductase. It is a regulatory enzyme. It regulates the cholesterol synthesis. And this is a cytosolic because we are going for cholesterol synthesis and it takes place in cytosol. Clear? Now we have 6-carbon compound, that is mevalonate. Next, it is converted to high Mbbs. M for mevalonate, I for isoprenoid units. And these are 5-carbon containing, right? And how will you remember them? Synthesis of HMG CoA is okay to all of you, just see this diagram. We have two molecules of acetyl CoA. They will form acetoacetyl CoA, right? And then another molecule acetyl CoA was added, we will get 3-hydroxy-3-methyl-butyryl CoA, that is our HMG CoA, right? And HMG CoA is precursor, that is our first step for synthesis of cholesterol, right? And after HMG CoA will form mevalonate, the second step mevalonate, right? And it requires the enzyme HMG CoA reductase. I have written it, and it is cytosolic, right? Because it takes place in cytosolic fraction of the cell, right? And now we are coming to this third step, that is, this was first, this was second, this is third, the formation of isoprenoid units, right? And these are all 5-carbon, and this is 6-carbon, this is 6-carbon. Ultimately we have to form C27H45OH. So ultimately we have to move to 27-carbon. We started from acetyl CoA, right? Then another acetyl CoA, then another acetyl CoA will form HMG CoA, and it is how many carbon? 6-carbon. From 6-carbon we are forming 5-carbon, that is isoprenoid units. The ultimate goal is to reach 27-carbon to form steroid ring complete for cholesterol, right? So let's see how will we make it. It is a 3-step reaction, right? And catalyzed by enzyme kinases. So we will form first IPP. Write down the short names as well. Isopentenyl pyrophosphate, right? And the second is DPP. It is dimethyl allyl pyrophosphate. Ultimately, that big diagram will come. I am making it short so you can remember it, right? So start from the mnemonic high miscellaneous, right? High for HMG CoA, M for mevalonate, I for isoprenoid units, S for squalene, and finally C for cholesterol, right? So we formed HMG CoA from two molecules of acetyl CoA, and another molecule acetyl CoA was added. We formed HMG CoA, that is 6-carbon. Then we formed mevalonate, 6-carbon. via HMG-CoA reductase reaction, and finally we got up to 5 carbon, that is isoprenoid units. And there are 2 isoprenoid units. One is isopentenyl pyrophosphate and another is DPP, dimethyl. Both are 5 carbon units. Right? Now, both are 5 carbons. Isopentenyl pyrophosphate and dimethyl pyrophosphate, both are 5 carbons. Right? Now, IPP and DPP condense to produce 10 carbon geranyl pyrophosphate. See, the isoprenoid units, they are all how many carbons? 5 carbons, right? We form 2 isoprenoid units, that is IPP and DPP, isopentenyl pyrophosphate and dimethyl pyrophosphate. Both are 5 carbons, right? And they will condense with IPP and DPP. 5 carbon plus 5 carbon. How many carbons you will get? 10 carbon, that is our geranyl pyrophosphate, that is our 10 carbon compound. Right? We started from simple compound, that is acetyl-CoA. Then another molecule acetyl-CoA was added, we get acetoacetyl-CoA. Then again acetyl-CoA, we got our HMG-CoA. High Mbbs, high for HMG-CoA. Then M for mevalonate, against the same number of carbon atoms will remain. Mbbs Mi, I for isoprenoid units. We form 2 isoprenoid units, that is isopentenyl pyrophosphate and dimethyl pyrophosphate. Both are 5 carbon, right? And these 2 will condense with each other. When they condense, 5 carbon plus 5 carbon will get our 10 carbon, that is our geranyl pyrophosphate, right? After that, we add, this is 10 carbon, we add another molecule of which? IPP, this isopentenyl pyrophosphate. So 10 carbon plus 5 carbon, we get 15 carbon compound. What is the name of this compound? 15 carbon, farnesyl pyrophosphate. Pyro or FPP. You can write FPP. Right? Starting from acetyl-CoA, we formed the first 6 carbon compound, that is HMG-CoA. Then it was converted to mevalonate, that is again 6 carbon. Then we formed 5 carbon units, that is known as isoprenoid units. And these isoprenoid units are 2, basically IPP and DPP, both are 5 carbons. They condense with each other to form 10 carbon, that is GPP or geranyl pyrophosphate. Then 10 carbon, again 1 isopentenyl pyrophosphate is added. 10 plus 5 is equal to 15 carbon, that is our farnesyl pyrophosphate, right? And the mnemonic is high Mbbs, M-I-S. S for squalene, and it is 30 carbon, right? How will you form squalene? This is 15 carbon compound, FCC. 15 carbon plus 15 carbon, that means 2 FPP, will get our 30 carbon squalene. 30 carbon compound, that is squalene. Now we have reached 30 carbon. The ultimate goal is to reach C27H45OH. So we have to reach, we have now excess carbon atoms, right? Now we'll have 19 enzymatic reactions, 19. But you will not remember them. I will just, there are total 19 enzymatic reactions that take place here. And finally, you will get our cholesterol, that is C27H45OH, right? To start with the mnemonic high Mbbs, high for HMG-CoA. How will you form HMG-CoA? From acetyl-CoA, another molecule acetoacetyl-CoA, then HMG-CoA, right? Now we have high component, high. Then next comes Miss M for mevalonate. Mevalonate is formed again 6 carbon by Tsmc co-reductase, that is a regulatory enzyme, right? Then from mevalonate we form isoprenogenous, that are 5 carbon compounds. They are two basically, one is isopentenyl pyrophosphate and second is dimethylallyl pyrophosphate. Both are 5 carbon. They will contain 5 carbon plus 5 carbon to 10 carbon, that is our garnesyl pyrophosphate or GPP. Then another 5 molecule carbon is added. Isopentenyl we get our 15 carbon, that is our farnesyl pyrophosphate. And then these two will combine. 15 plus 15 is equal to 30. 30 is your squalene. High Miss S for squalene. And final component is C, that is cholesterol, that is contains C27H45OH. And from this step, there are 19 enzymatic reactions. I will basically tell you 3-4 major, and finally we get the cholesterol, right? Let's move to diagram. Please get this PPT and get screenshot. So IPP and DP condense to form 10 carbon garnesyl pyrophosphate. Then 10 carbon plus 5 carbon will form 15 carbon, that is our farnesyl pyrophosphate. Then 15 carbon plus 15 carbon, 2 molecules FPP will form 30 carbon, that is squalene. Clear to all of you now? Now, final is conversion of squalene to cholesterol, right? It undergoes hydroxylation and NADPH gets converted to lanosterol. These are short steps. If you can remember it, otherwise you can write down the state steps, right? And lanosterol will undergo 19 enzymatic reactions, I told you. After 30 carbons, we get squalene. As squalene is converted to lanosterol, right? And after that 19 reactions take place, the basic are reduction of carbon atom from 30 to 27, I told. The formula of cholesterol is C27, not 30, right? So 3 carbons are reduced. Removal of 2 methyl groups from C4 and 1 from C14. Shift of double bonds from C8 to C5. And reduction in double bonds of C24 and 25. And finally we will get penultimate product, that is 7-dehydrocholesterol, and which finally will yield our cholesterol. कितना बोरिंग है कि नहीं है? Many of you are yawning in the class. And this is the overall. So beautiful. So again, see acetyl CoA. Now it is made simple by me, na? Just see, you can remember in the whole picture. Acetyl CoA plus acetyl CoA, 2 molecules will get acetoacetyl CoA. Then again acetyl CoA is added, will get HMG CoA, right? And then HMG co-reductase will form mevalonate. High Miss M for mevalonate. Then kinase reaction and mevalonate से क्या बनेगा? Isopenoid units Mi, or these are pyrophosphate, and will get phosphomevalonate and isopentenyl. This 5 carbon is formed, 5 carbon, and 5 carbon again dimethyl, like these two, isopentenyl and DPP, right? So we get IPP and DPP. IPP was 5 carbon, isopenoid units, isopentenyl pyrophosphate and dimethylallyl pyrophosphate. This is 5 carbon and this is 5 carbon. 5 carbon plus 5 carbon, how many? 10. 10 carbon is geranyl pyrophosphate, right? And 10 carbon again, 5 is added by IPP, will get our 15 carbon, that is our farnesyl pyrophosphate. And 15 carbon plus 15 carbon will get our squalene, 2F farnesyl pyrophosphate, and will get our squalene, right? And this is squalene is written as circular form, and it is converted to 30 carbon, that is lanosterol, and finally via 19 reactions we will get our cholesterol, C27H45OH. It is finally formed. Please revise it. It is in the syllabus, right? Anybody who can repeat after me, who has learned very well? Miss Sinha and Miss Anushka, no noise at the back. Yeah, no noise. Yes. Clear to all of you or any doubts? Now she is running in the class. Gayatri, इसको गायत्री सुनाओ, क्या करें इसका? It's clear to all of you now. Energy cost, how many ATP are consumed? Complex energy expensive process, ATP is consumed only in the step that convert malonate to isoprenoid units. The second and third step require ATP, right? How will you form? ATP is consumed in the step that would malonate to isoprenoid units. 3 ATP molecules are used to create 6-membered isoprenoid, right? And 3 into 6 will be total 18 ATP are used for ATP synthesis, right? Or for cholesterol synthesis. So this is the complete structure. Cyclopentanoperhydro, 1 6-membered ring, again 6-membered, again 6-membered, 5-membered and a side chain. C27, H45 or OH or C27, H46 O only, right? And these are the steps starting from 2-carbon acetyl CoA, high Miss, HMG CoA, high Miss, mevalonate, Mi Miss, isoprenoid units, 5-carbon, Miss MIS, 30 carbon squalene, 30 carbon lanocidol, isomeric form, and finally 27 cholesterol. It's okay. And now we'll start with the regulation. How will you regulate cholesterol? लिपिड प्रोफाइल घर में करवाते हैं सारे ना पेरेंट्स, तुम भी करवाते होगे. किसी का कोलेस्ट्रॉल ज़्यादा आता है, किसी का कम आता है. इसका ज़्यादा है, इसका ज़्यादा है. Why is this problem? Some have high cholesterol levels and some have low cholesterol levels. Regulation is not okay, but dietary lifestyle is very important as well, right? So let's see. The main enzyme is for regulation is HMG CoA reductase. This is the major enzyme. Conversion of HMG CoA to mevalonate, the first step, and the enzyme is HMG CoA reductase. And this is the major enzyme that plays the role. So how will you first regulation is feedback regulation. Feedback. See the name feedback. Suppose you already have good levels of cholesterol. Feedback. Right? When you excess cholesterol, the end product will stimulate to stop the synthesis of excess cholesterol, right? And it acts on which enzyme? I told you HMG CoA reductase, right? The end product is cholesterol and it controls its own synthesis by feedback mechanism, right? This is achieved by decreasing the transcription of gene responsible for HMG CoA reductase. When the body has enough cholesterol, the levels are good, right? And the steps required, the first step is the enzyme is required, that is HMG CoA reductase. So it will decrease the gene responsible for synthesis of HMG CoA reductase. It will decrease its transcription, decrease the transcription of this enzyme. So it will not be formed and cholesterol synthesis will decrease all. Okay, so this is feedback control. The second control is this is feedback. Excess cholesterol is there. It will inhibit the transcription of this gene that will stop the synthesis of HMG CoA reductase. Right? Now, second is hormonal regulation. You have seen or talked to your someone, elder brother or sister, जिनको थायराइड की प्रॉब्लम होती है, उनका कोलेस्ट्रॉल बढ़ जाता है. In the family, those who are hypothyroid, their cholesterol level is almost or always high because of this regulation. right? I told you HMG-CoA, first thing is that it is cytosolic and mitochondrial. Cytosolic will act in C for cholesterol synthesis, it will act in cytosol. Another one is mitochondrial, that will influence ketone body synthesis, right? Now it has also two forms again, HMG-CoA, one is active and another is inactive, right? And one is phosphorylated and another is non-phosphorylated. We'll use this mechanism here. See, two interconvertible forms are there, and D-phospho form HMG-CoA is active, and while phosphorylated form is less active, right? HMG-CoA, one is active form and one HMG-CoA that is inactive form. Active form is there, how you remember it? It is D-phospho, and this is phosphorylated, right? It is converted to one and another. See, the hormone influence through a cyclic AMP by series of reaction that is comparable with glycogen synthesis reaction as well, right? And the next effect is glucagon and glucocorticoids, they favor the synthesis formation of active HMG-CoA. When you have excess levels of, one is glucagon and the second is glucocorticoids. What they favor? They favor the synthesis of HMG-CoA that is active. Favor the formation of and decrease cholesterol inactive. So it will decrease and cholesterol synthesis will finally decrease. Someone is there? And other two, insulin and thyroxine, they increase cholesterol production. They decrease, and insulin and thyroxine, what will happen? They will enhance the formation or increase the formation. Insulin can enhance the formation of cholesterol. What does it indicate? Simple trick for weight loss, I am telling you now. Simple trick, those who are overweight, they can try it and it will work wonders. When insulin levels are high, in the body, when glucose levels are high in the body, right? But when insulin levels keep high, what will they favor? They will favor cholesterol synthesis, right? So simply you eat anything, but stop eating sweets. It will work because no glucose is there. If glucose is not there, there will be no insulin spike. So there will be no insulin. And no insulin, no cholesterol synthesis. Cholesterol will build fat in the body. So simple thing is that you stop eating sweet. Eat anything, non-veg, carbo, anything, but stop eating sweet. If you will not eat sweet things, glucose will fall. Glucose will remain static, right? So insulin spikes will not be there. When insulin levels are not high, the cholesterol synthesis will not go for it. Body will not go for cholesterol synthesis. What will the body will go for then? Cholesterol breakdown. Breakdown में क्या होगा? Fat breakdown होगी, ketone bodies बनेगी, or you will lose weight. Simple thing is that, right? Now the hormonal regulation is okay with you. Third is inhibition by drugs. Statin दे दो statin. Everybody you visit a physician, he will prescribe you some statins, lovastatin, statins, drugs, right? How will they act? They will act on the same enzyme, that is our HMG-CoA reductase. How? The major two drugs are copectin and lovastatin. They are fungal products. And how they act? They used to decrease cholesterol levels in patients with hypercholesterolemia, right? They are competitive inhibitors of enzyme HMG-CoA reductase, right? We have seen competitive inhibition in which chapter? Enzymes. We are using two chapters here. One is enzymes, competitive inhibition, right? And it is acting on which enzyme? That is our second enzyme, that is our HMG CoA reductase. The cholesterol metabolism is regulated to this enzyme only. Both are competitive inhibitors of HMG CoA reductase and reduce cholesterol synthesis by 50 to 60%. Clear? And HMG CoA reductase is also inhibited by bile acids. And fasting also boosts the activity of this enzyme. If you fast, what will happen? It is told by some senior citizens and older people that, or yogi monks, they tell you fast, you will lose weight. How? Because it will decrease the activity of HMG CoA reductase. So everything is linked with biochemistry, right? And cortisol synthesis breakdown, it is linked with one enzyme, one and one superhero, that is our HMG CoA reductase. So that almost finished, and in the next class we will discuss breakdown and lifestyle modification to maintain it. Fate of cholesterol next में करेंगे. And anybody who has doubts, or I will revise बोलो, or I will ask questions. Server Adil, are you ready? Hormonal वाला. These are four basic hormones. One is glucagon, glucocorticoids, insulin, and thyroxine. These two play separate role. These two play separate role, right? Insulin, thyroxine. What will they favor? I told you when you will eat sweets, the glucose will increase. The glucose will increase. What will the body secrete to decrease it? Insulin. And insulin decreases finally, right? And hypothyroid patients, the case is discussed, right? Increase cholesterol production, right? And the glucagon and glucocorticoids, they decrease cholesterol synthesis, right? It's clear. Now the enzyme is, major enzyme is HMG CoA reductase, right? It has two forms. One is active and other is non-active or less active. An active form is dephosphorylated and non-active is phosphorylate. So it will regulate by this. It will convert it into either active form or inactive form by phosphorylating it or dephosphorylating. These two, glucagon and glucocorticoids, they decrease, right? They decrease, that means it will inactivate this enzyme. And insulin and thyroxine, they increase, that means they will activate this HMG CoA reductase. An active form is, so it will remove phosphate from it. It will become active, and active will lead to cholesterol synthesis. Let's see one diagram I missed. This is HMG CoA reductase. The first mechanism was feedback. When we have excess cholesterol in the body, cholesterol will stop the transcription of this gene of HMG CoA reductase and it will inhibit the synthesis. Second, glucagon and glucocorticoid, they will inhibit this enzyme HMG CoA reductase, right? So HMG CoA reductase will, cholesterol synthesis decrease हो गई, and this insulin thyroxine, they will increase the activity of this enzyme, so it will form cholesterol. And these two are compacting lowers, their drugs, They are competitive inhibitors. So it will inhibit this enzyme HMG co-reductase. So that finishes